Good evening, Assalamu alaikum. Good evening. It gives me great pleasure and honor to welcome you all on behalf of Sheikh Hamdan Award of Medical Science to this evening scientific meeting titled A Common Lesson of a Rare Disease. Over the next 45 minutes, Professor Frederick Kaplan will show us how is the key to the closet can be the key to the kingdom. But before we go to the main event of this evening, I would like to especially welcome the Board of Trustees members who are continuously supporting the award virtual activity. Secondly, I would like to give you a brief account on Sheikh Hamdan bin Rashid Award of Medical Science. It was established in 1999. It's the vision of His Highness belated Sheikh Hamdan bin Rashid Al Maktoum that comprehensive healthcare system relies on the latest scientific advancement and people empowerment. For years, the award has been the driving force to support medical field in general. It collaborates with academia and healthcare sector to honor excellency and fund researchers. Indeed, these efforts have been paid off well, as two of the Grand Hamdan Award winners were awarded Nobel Prize in Physiology in 2010, Sir Rupert Edward, and in 2020, Dr. Harvey Alter. With that note, we'll move to Professor Kaplan, who was awarded Grand Hamdan International Prize in Medicine in 2018. Professor Kaplan, he's an international icon in the field of orthopedic molecular medicine and considered the leading expert of world-leading expert on genetic disorder of heterotopic ossification. He is the chief of the division of orthopedic molecular medicine at the University of Pennsylvania. He directs the Center for Research for Fibrodysplasia Ossificans Progressiva and Related Disorder. In 1997, Professor Kaplan was awarded the first endowed chair in the United States for orthopedic molecular medicine. Newsweek journal named Professor Kaplan to be one of the top 15 who makes America great in 2006. And in 2011, Professor Kaplan was elected to the National Academy of Medicine. And Professor Kaplan was honored with National Organization of Rare Disorder Real Impact Award in 2017. And of course, he's the Grand Award winner of Sheikh Hamdan Award for International Award for Medical Science in 2018. With that, without further ado, I will leave the mic for Professor Kaplan to present his speech on a common lesson of rare disease. Professor Kaplan, the mic is yours.
Thank you very much. Distinguished members of the Royal Academy, Dr. Jamal, ladies and gentlemen, I want to thank you very much for inviting me back to Dubai to deliver this encore address of the Grand Hamdan Award and for the extremely kind and generous introduction. The title of today's webinar is "The Key to the Closet is the Key to the Kingdom." And it starts in London in 1657, 364 years ago, when a letter arrived at the home of Dr. William Harvey in the last year of his life. In fact, in the last week of his life. Dr. Harvey is arguably one of the most famous doctors in all of medicine, and he discovered the circulation of the blood. And this letter was from Dr. Blackveld from Harlem, and the letter was dated April 24th, 1657. It was 364 years ago. And Dr. Blackveld asked Dr. Harvey to come across the English Channel and see a patient who had a rare bladder condition, a genetic, probably a genetic condition of the bladder, But Dr. Harvey picked up his pen immediately and wrote back to Dr. Blackfeld that he couldn't go, that he was very ill, and that he could not make the journey across the English Channel. But then he wrote probably some of the most famous words in all of medicine. And he said to Dr. Blackfeld that it's very important that somebody investigate this. And he said, and he wrote in the letter, and this is the last letter he wrote in his life, about a week before he died. He said, nature is nowhere accustomed more openly to display her secret mysteries than in cases where she shows traces of her workings apart from the beaten path. Nor is there any better way to advance the proper practice of medicine than to give our minds to the discovery of the usual laws of nature by the careful investigation of rarer forms of disease. For it has been found in almost all things, he wrote, that what is useful or applicable is hardly perceived unless we are deprived of them, or they become deranged in some way. And the rationale for this was described by Fishman and Porter in an article in Nature in 2005. The rationale for extrapolating from rare genetic diseases, they said, to common sporadic illnesses is that nature is conservative. This is a safe bet, they said, and there would be immense immediate benefit in tackling the rare diseases themselves. This approach could have a salutary side effect, a refocusing of drug research on neglected diseases. As Richard Feynman, Nobel laureate in physics, said, nature uses only the longest threads to weave her patterns, so each small piece of her fabric reveals the organization of the entire tapestry. In other words, the key to the closet is the key to the kingdom. Rare diseases are a worldwide health care challenge. They affect approximately 6% of the population of Western societies and compromise about 10,000 disorders. More than 80% have an identified genetic component, and most are disabling. The closet, the key, and the kingdom. Let's look at a few genetic diseases. The closet, let's say familiar hypercholesterolemia that you're probably all familiar with. The key is the LDL receptor. And what did it unlock? It unlocked the kingdom of cholesterol metabolism. Leschnayan syndrome, a very rare condition that you see in metabolism and rheumatology, is due to mutations of the HGPRT gene, hyperxanthine guanine phosphoryl and unlock the kingdom of purine metabolism. Progeria, an extremely uncommon and extremely disabling disease of childhood, is due to lamin A mutations and unlock the kingdom of nuclear envelope regulation. Hemophilia, due to factor VIII deficiency, unlock the kingdom of blood clotting disorders. Malignant hyperthermia, due to the ryanodine receptor mutations, unlock calcium channelopathies. And male infertility in one patient who had 5-alpha reductase deficiency that led to the prostate not forming, led to drugs that deal with prostate disorders and hypertrophic disorders of the prostate. Achondroplasia, due to the FGFR3 mutations, led to understanding of the growth plate. Duchenne's muscular dystrophy, mutations of dystrophin, led to unlocking the kingdom of muscle physiology. McCune-Albright, pseudohypoparathyroidism type 1, Albright's hereditary osteodystrophy, osteomycutis, and POH, all part of a spectrum of disorders, all due to GNAS mutations, led to understanding and unlocking the kingdom of G protein coupled receptors. And FOP, which we'll talk about today, fibrodysplasia ossificans progressiva, the key is activin receptor 1A. It led to understanding and unlocking the kingdom of BMP signaling pathways. Myositis ossificans is a common condition, but the progressive form is extremely rare. It's an ultra rare disease, and this is a copy of a notebook from medical school more than 45 years ago. And I wrote then, I had no idea that it would become the the focus of my medical career, that the progressive disorder was of a yet unidentified etiology. The clinical features of FOP, or fibrodysplasia ossificans progressiva, as I described in my Grand Hamden lecture, are malformations of the great toes. There are other malformations of joints, but every patient with a classic condition, about 97% of patients who have FOP have abnormalities, malformations of the great toe. And then probably around the ages of 2 to 5, painful swellings that look like tumors seize the skeletal muscles, tendons, ligaments, fascia, aponeurosis, and turn them into bone through an endochondral process. Any attempt to remove this heterotopic bone leads to explosive formations of new bone and even minor bumps and bruises, injections into the jaw for dental work, intramuscular immunizations, biopsies, etc., can cause progression of FOP. We see here on a CT scan of a patient that ribbons, sheets, and plates of bone form an FOP, in a sense, forming a second skeleton. And as we can see here, in a patient, we can see how disabling this condition is. This is a young man who's about 30 years of age, and you can see in this video how the heterotopic bone has completely replaced and locked all of his joints. In 2006, Meichi Su from our lab discovered the gene that causes FOP. She was the first to see this. It's a heterozygous activating mutation. It causes mild constitutive activity in the basal state, and with inflammatory triggers leads to explosive new bone formation. So one misspelled letter in 6 billion causes FOP, and this is the cause of FOP in every single individual who has classic FOP. 97% of individuals, the 3% of individuals that have variant FOP have a variance, other mutations, I should say, in this gene. How we found this gene was described in my Grand Hamden lecture. I'm not going to go into that, but suffice it to say that we published a paper in 2006 in Nature Genetics that described a recurrent mutation in the BMP type 1 receptor, ACBR1, and it causes inherited and sporadic fibrodysplasia ossificans progressiva. And the following week, the journal Science published an editorial that said the bone disease gene was finally discovered, it was finally found. And this unlocked an entire pathway. It unlocked the BMP pathway, the bone morphogenetic protein pathway. The mutation is in a receptor, it's in the transmembrane, the intracellular portion of the receptor, I should say, not the transmembrane, in the GS domain. It causes a mild constitutive activation, and when it's triggered by inflammation, causes dramatic inflammatory signals to go from the receptor to the target genes, which cause heterotopic ossification in the responsive tissues. And 364 years after Dr. Harvey wrote his letter to Dr. Blackfield saying that he couldn't come, but rare diseases are important, the New York Times in New York published a lead article in their medical journal, in their medical section. It said, finally, with genetic discovery, hope. And that's the key word, hope. Hope for escape from a prison of bone. And here you see a picture of my patient, Hayden Fife from Sausalito, California. And clearly in 2006, the FOP gene discovery, after the FOP gene discovery, there was a remarkable rise in the interest of the pharmaceutical companies. And the targets, of course, for FOP are soft tissue injury or spontaneous flare-up, inflammation, and tissue hypoxia, which we'll get into. the ligands which act on the receptors, the ligands BMP4 and activin A, which act on the ACVR1 mutant receptor to cause heterotopic ossification, and this discovery unleashed the potential treatments, which are injury prevention, of course, immunosuppression, anti-inflammatory drugs and mast cell inhibitors, HIF1 alpha inhibitors, monoclonal antibodies against activin A and BMP4, signal transduction inhibitors, palovarotene or the retinoic acid receptor gamma agonist, and again, and eventually, when there's a combination of drugs that allows for this receptor to be quiet, we can consider surgical removal of bone. And I will mention that immunosuppression, anti-inflammatory drugs, mast cell inhibitors, HIF1 alpha inhibitors, monoclonal antibodies, and signal transduction inhibitors, as well as retinoid receptor agonists, all unleashed by the discovery of the ACVR1 gene, are now in clinical trials for FOP. The dysregulated signaling pathway in dysregulating BMP signaling pathway involves pulmonary hypertension, which affects the lungs. It's due to mutations in BMPR2, hemorrhagic telangiectasia, which is due to a similar gene receptor, ALK1, which affects the blood vessels, juvenile polyposis syndrome, which affects the downstream signaling pathways, affects the bowel, pancreatic cancer, also affects the downstream signaling pathways. Diffuse idiopathic pontine glioma, which affects the CNS, is due to ACVR, sporadic and somatic mutations of ACVR1, and obviously FOP, which affects the skeletal system. Now, the dysregulated BMP signaling pathway and all of these conditions informs us about the side effects, the off-target effects, and but especially the collateral effects of drugs. And this helps us understand and monitor what some of those side effects will be if we inhibit the BMP signaling pathway or by our therapeutic measures. So we're looking for pathology in the lungs, in the blood vessels, in the bowel, in the pancreas, and the CNS as well for in our therapies for FOP. Donald Coffey wrote in an article in Nature Medicine in 1998 that the self-organization of molecules into complex networks are a feature of emergent properties. In such systems, small effects like a single amino acid substitution, like we see in FOP and ACVR1, in a receptor that is part of a signaling network, can have very large and unexpected consequences with new properties not possessed by the original system. And these emergent properties can manifest as disease. Such insights can advance our understanding of biological function beyond genomic sequence into the realm of pathophysiology. And in the last several years, we've discovered a number of emergent properties of the gene that we discovered. We wouldn't have discovered these emergent properties if we didn't have the gene. So this, in a sense, is a hook. It's a hook to understand the emergent properties. And so we're looking inward, not outward, but inward at FOP. And these emergent properties that I'll describe are that mutant ACVR1 is sensitive to new ligands. Who would have known this? Mutant ACVR1 signaling is amplified by cellular hypoxia. Mutant ACVR1 signaling is directly linked to the innate immune system activation. Amazing. Mutant ACVR1 signaling is amplified at increased temperatures. Mutant ACVR1 signaling impairs joint formation and joint survival. And mutant ACVR1 orchestrates different phenotypes through different progenitor cells. Let's take a look at some of these emergent properties. Mutant ACVR1 is sensitive to new ligands. The old school of thought was that the faucet, which represents ACVR1, and the water coming out of the faucet represents the signal transduction that leads to heterotopic bone, or that leads to bone. We thought that the bone morphogenetic proteins turn on this faucet when you want bone to form, and when you don't want bone to form, this faucet is turned off. The new school says that the BMPs turn the faucet on, but activin A turns the faucet off. And normally, in development, this faucet is a bimodal sort of a faucet. It's a bimodal switch. It can be turned on, it can be turned off. And this was discovered by scientists at Regeneron Pharmaceuticals. And they discovered in FOP, this is the old school, we thought that the BMPs turn the faucet on, the drippy faucet that leads to the malformations of the toes in embryogenesis, the osteochondromas, the congenital malformations that we see, and the bone morphogenic proteins turn it on. But the scientists from Regeneron showed that the activin A actually acts to turn the faucet on, not to turn the mutant faucet on, not to turn the mutant faucet off. So in other words, the mutation causes a brake pedal to turn into an accelerator pedal. There's no other example in nature where a ligand acts differently on the mutant receptor than it does on the normal receptor. Now, we don't know the reason for this yet, but we do know that it's a fact. And the best example of this is activin A, which is a ligand that acts on to activate the mutant receptor, inhibits BMP pathway signaling through wild-type ACVR1. So we see here in the FOP mouse, in an FOP mouse that has all the features of FOP, it has both the mutant and the wild-type receptor, and it forms heterotopic bone. But when we remove the wild-type receptor, we get a tremendous amount more heterotopic bone. This showing that the mutant receptor actually forms heterotopic bone, signals through activin A to stimulate heterotopic bone, but the wild-type receptor acts as a brake, and if you remove the wild-type receptor, you get even more and dramatic activation of bone formation. Another emergent property is mutant ACVR1 signaling is directly linked to the innate immune system activation. We showed several years ago that a gene called exit, evolutionarily conserved signal transduction in the toll pathway links TLR, toll-like receptor signaling, and BMP signaling in FOP connective tissue progenitor cells. And this study provides a direct link between the innate immune system activation and dysregulated BMP signaling in FOP. And we see here that injury can cause PAMs and DAMs to activate the toll-like receptors, which act through NF-kappa B and IRF3 to cause inflammation and hypoxia. They also induce bone morphogenic proteins, and they indirectly activate the macrophages and the mast cells to produce activin A, which acts on this receptor. But it's very, very important to recognize, and we had no idea that this is the case, that a gene called evolutionarily conserved signal transduction in the toll pathway, which has been present since the time of Drosophila, so for 500 million years, links the inflammatory pathway to the BMP pathway intracellularly and allows these two pathways to communicate. And this is probably one of the most important pathways for activation of FOP that we showed. And so who knew, but FOP, there is a clue from the fly, and there's a clue. This is the first paper we wrote back in 1991, but we had no idea then, this is before the discovery of the gene, that the ACVR1 pathway, the bone morphogenic protein pathway, would be linked to inflammatory signaling. Another emergent property that we discovered was mutant ACVR1 signaling is amplified by cellular hypoxia. And there's a paper that we wrote that showed that cellular hypoxia promotes heterotopic ossification by amplifying BMP signaling. So we have here tissue injury causes inflammation, and hypoxia, as Dr. Semenza, who won the Nobel Prize for discovery of HIF-1 alpha, had described, that hypoxia is sort of the flip side of the coin of inflammation. And HIF-1 alpha, the protein HIF-1 alpha is activated. And we saw in FOP that HIF-1 alpha was directly responsible for dramatic increased signaling in FOP. So what is the relationship between hypoxia and inflammation in HIF-1 alpha, and why does HIF-1 alpha cause a dramatic increase in BMP signaling? Well, a clue came from cancer. Many cancers are caused by activating mutations in growth factor receptors, and signaling in cancers are dramatically amplified by hypoxia. See if I can advance this slide here. Let's direct our attention to the bottom of the slide, where hypoxia causes HIF-1 alpha activation. It causes HIF-1 alpha to the protein to be available. And we know that HIF-1 alpha can turn on many genes, but it turns off several genes. And it turns off a very important gene called ribaptin-5. And ribaptin-5 complexes with RAB5, and these escort receptors to the endosomes for degradation. Well, obviously in cancer, if there's for example, if there's a activating mutation of the EGF receptor, and ribaptin-5 isn't made, then these receptors stay on the cell membrane and signal for a long period of time. As analogy, BMP or activin A, which induces ACVR1, we showed that with these lesions that occur, these dramatic inflammatory lesions are incredibly hypoxic. And we showed that. We showed that these lesions are hypoxic in both humans and in mice. And HIF-1 alpha is activated, and ribaptin-5 is turned off. Well, if ribaptin-5 is turned off, these receptors that signal cannot be escorted to the early endosomes, just like in cancer, and they're not degraded. Well, very interestingly, the BMP receptor, ACVR1, not only signals on the cell membrane, but it signals dramatically in the early endosomes. So this is a dramatic example of amplification of BMP signaling that occurs in hypoxia. And we showed that hypoxia can be the flip side of inflammation. So inhibition of ribaptin-5, there's prolonged endosomal retention of ACVR1, there's increased BMP signaling, and heterotopic ossification. And imatinib, which has many effects, it's an anti-cancer drug, but one of its side effects, or one of its off-target effects, I should say, is on target for FOP, and it blocks HIF-1 alpha. And we're going to start a clinical trial using imatinib in very young children who have FOP, in children at an age where we can't use or we can't enroll them yet in the trials for signal transduction inhibitors. So this is something that went from the lab directly to the clinic. So injury triggers inflammation and hypoxia, probably through the innate pathway, and it causes a light bulb to be turned into an atom bomb, dramatic amplification, which occurs in FOP. Another emerging property which was discovered by us in the last several years is that mutant ACVR1, which is responsible for FOP, mutant ACVR1 signaling is amplified at increased temperatures. And it's very interesting to note that FOP doesn't progress randomly. It progresses in certain anatomic gradients. So children are often affected in the back first, and then in the front of the body. So the dorsal regions are affected before the ventral regions, the cranial regions before the caudal regions, and the axial before the appendicular. And so here we see a young child with flare-ups, dramatic swellings that lead to heterotopic bone, inflammatory swellings with dramatic hypoxia, and they affect the back. And this is common. And it dawned on us that that the pattern of FOP progression from dorsal to ventral, from axial to appendicular, from cranial to caudal, from proximal to distal, follows the patterns of temperature in the body, not just surface temperature, but deep muscle temperatures as well. And this is an example from normal individuals from thermography. And in areas where there's increased heat, such as the back and the neck, there are deposits of brown fat, which are incredibly thermogenic. And a light bulb went off. And we said ACVR1, which encodes ALK2, which is the name of the receptor. ALK2 is a transmembrane receptor. ALK2 is activin-like kinase 2, which anything that ends in ACE is an enzyme. ALK2 is also a transmembrane enzyme. And enzymes are heat-sensitive catalysts. And the ALK2 enzyme is a heat-sensitive catalyst of SMAD1/5 signaling pathway. And as you can see here, we all learned in chemistry that that there's an optimum temperature for enzyme activity, and it's obviously at the physiologic temperature of peak body temperature, of core body temperature, I should say, which is seen in the areas that are first affected by FOP. And this is an FOP child held by the mother. You can see the child here, and you can see the hottest areas in the back of the child. And this is a 4-year-old child with FOP, and you can see that again, the temperatures reproduce the patterns of bone formation that we see. So we decided to isolate stem cells from mice with FOP, and we subjected them to different temperatures, and we found that at all temperatures, the signaling was greater in FOP cells than in normal cells. We found that the downstream signaling from FOP was highest at peak body temperature, and we found that the differentiation of cartilage and bone was higher in FOP cells at all temperatures than in normal cells, and you can see that here. And the differentiation of into cartilage and into bone was higher in FOP cells than in all normal cells. So in summary, the anatomic distribution of heterotopic ossification in FOP might be due in part to the dysregulated temperature response of the FOP gene ACVR1 receptor kinase, the mutant receptor kinase, relative to wild type receptor that meets or exceeds the threshold for signaling output in responsive connective tissue progenitor cells as a function of local temperature. So, and while the association of physiologic body temperatures with anatomic patterns of HO and FOP does not prove causality, temperature can be added to a growing list of microenvironmental factors that likely influence the initiation of FOP lesions and subsequent FOP. And this was published in an article entitled "Spatial Patterns of Heterotopic Ossification in FOP Correlate with Anatomic Temperature Gradients." Another emergent property that we discovered in the last several years was mutant ACVR1 signaling in FOP impairs joint formation and joint survival. A comprehensive understanding of FOP requires as complete an understanding of skeletal patterning and morphogenesis as it does of heterotopic ossification. And Victor Hugo, the novelist, said that by measuring the toe, we estimate the giant. And here you see a skeletal preparation of the malformed toe in FOP. This is not just a bunion. This is a true malformation of the metacarpophalangeal joint. There are malformations of other joints as well, as we'll see. And we see here that there are, not due to heterotopic ossification, but due to intra-articular fusion, there are fusions of the facet joints of the cervical, thoracic, and lumbar vertebra. Shown here are the thoracic and lumbar vertebra. There are, in the second panel, There are fusions of the costovertebral joints where the ribs join the spine. We all know that there are 12 ribs on each side. Each rib has 3 articulations with a vertebra. That's 36 joints. 36 times 2, there's 72 joints in the body that form and then fuse prematurely due to intra-articular fusions due to abnormalities in these joints. Here is the knee joint that's fused due to an intra-articular fusion, and the hip joint that's fused, not due to heterotopic bone. There is some heterotopic bone, but it's fused due to intra-articular fusions. And Will Towler, who was a graduate student in our lab, published 3 incredible papers showing that dysregulated BMP signaling through ACVR1 impairs joint formation development in FOP. And basically here, what we have is normal compared to FOP. We have normal ACVR1 signaling through the canonical BMP pathway, and mutant ACVR1 signaling, dysregulated BMP signaling through the FOP pathway. So dramatic increased BMP signaling. In the normal pathway, the BMP pathway inhibits a pathway called Wnt. And Wnt is very important in joint formation. And this is very important in the formation of the joints. And so our joints all form from one ball of cartilage. The limbs form from one piece of cartilage. And then the joints form by inhibition of the BMP pathway where the prospective joint will form. And Will showed that with increased BMP signaling, you have an increased, dramatically increased inhibition of Wnt, which is essential for joint formation, and the joints don't form properly. Now, certain joints are more susceptible, like the great toe, but other small joints as well, as we've discovered, the facet joints of all the entire spine, the costovertebral joints, the hip joint, the knee joint, many joints as well do not form properly in FOP. Another emergent property that we discovered is mutant ACVR1 orchestrates different phenotypes through different progenitor cells. And we discovered that GDF5, the ACVR1 gene expressed in GDF5 cells that Will showed is responsible for the joint abnormalities. So increased signaling through these progenitor cells that are very important in embryogenesis to make up all the joints of our body. Certain joints are more sensitive to BMP signaling and inhibition of Wnt signaling than others. But if you have over suppression of Wnt signaling, you don't get joints to form properly. And if you do get them to form, you get them to fuse prematurely. And in this next slide, we showed that PRRX cells, cells that are expressed around the growth plates of bones, cells that express the PRRX gene around the growth plate of bones are responsible for the osteochondromas in FOP. And most importantly, the fibroadipogenic cells in the muscles, tendons, ligaments, fascia, and aponeurosis, the fibroadipogenic progenitor cells. So just let's take a look at the muscle. It's not the muscle cells themselves that turn to bone. It's not the satellite cells that regenerate muscle that turn to bone. It's the fibrous cells in the muscles that that explode into dramatic fibroproliferative lesions that are incredibly hypoxic, and that lead to chondrogenesis and form the heterotopic bone. So different progenitor cells are responsible for different phenotypes in FOP. The phenotype that we see in embryogenesis, the phenotype we see in childhood, and the phenotype we see in adulthood. And so the key to the closet, ACVR1, is the key to the castle of FOP. This is sort of looking inward due to these emergent properties. And this key has unlocked doors to cancer, to developmental biology, to enzymology, as we've shown, to evolutionary biology. We would have never been able to discover that if we didn't have the ACVR1 gene, that hook that enabled us to look inward. It has unlocked the doors to hypoxia, to inflammation, to the innate immune system, to joint development and degeneration, to pharmaceuticals for FOP, and to the stem cells that are responsible for FOP. Carl Zimmer wrote in an article in the journal Atlantic Monthly, a tiny percentage of the world's population suffers from FOP, which locks its victims in cages of superfluous bone. For centuries, these patients were dismissed as a lost cause. But recent genetic and technological advances have propelled researchers worldwide towards an understanding of this disease that may transform the lives not just of people who suffer from it, but of those afflicted by much more common ailments. Rare diseases, it turns out, are more relevant than we ever imagined. Heterotopic ossification is a common condition, and FOP is just the tip of the iceberg. Where does heterotopic ossification occur commonly? It occurs commonly after joint replacements. Many of you or orthopedic surgeons know that hip replacements, especially in certain individuals, can lead to heterotopic ossification. Sometimes this can ruin the joint replacements. Head injuries, central nervous system injuries lead to heterotopic ossification. And we don't know why yet, but the BMP pathway is involved. But it may not be the same BMP pathway, the same abnormality in the BMP pathway that causes FOP, but it focuses attention on the BMP pathway. Spinal cord injuries, again, central nervous system injuries can cause heterotopic ossification. Burns through inflammatory disorders can cause heterotopic ossification. Blast injuries can cause heterotopic ossification. And the soldiers in the wars that returned from Iraq and Afghanistan that had traumatic blast injuries had a tremendous number of them had heterotopic ossification. And we see heterotopic ossification in vascular disease. It's not just calcification of the arteries, it's ossification, about 12 to 13%. And we discovered many, many years ago that valvular heart disease was not just due to hardening of the valves due to calcification, but actually 12 to 13% had ossification. And this led to the discovery of ossification in vascular disease as well. And this was published in a paper, Bone Formation and Inflammation in Cardiac Valves. Now, FOP patients do not get a bone formation in their cardiac valves, and they don't get intravascular ossification. But the same process occurs histologically. You see endochondral bone formation. You see inflammatory and induction of heterotopic bone formation in the case of valvular disease and vascular disease. So FOP has led to these discoveries as well. And I wonder if there's a new horizon to harness the explosive heterotopic skeletal formation of FOP to create bone for those who need it. So if we can create the renegade runaway bone formation of FOP and tame it, turn an atom bomb, an atomic explosion back into a light bulb, can we create bone for those who need it in fracture healing, in spinal fusions, in congenital defects, in traumatic loss of skeletal elements? And can we inhibit the FOP signaling pathway to inhibit joint degeneration? And this will be very interesting to see in the clinical trials. Clearly, the primary endpoint of many of the clinical trials that are ongoing and that will come in the next year or so will be to inhibit heterotopic bone. But we'll also be interested to see whether they extend the life of some of these joints by inhibiting the BMP pathway. And the question is this a fait accompli in embryogenesis, or is it possible to inhibit the BMP pathway postnatally and extend the life of some of these joints? So this we'll see. We don't know the answer to these questions yet, but I can tell you that engineers are looking at FOP as an example and trying to harness that for fracture healing, for spinal fusions, for congenital defects, and traumatic loss of skeletal elements. And then this, we wonder whether it's possible to inhibit the FOP signaling pathway to slow joint degeneration. So FOP is relevant to every condition that affects the formation of bones, and especially the formation of the skeleton. So FOP is not just a bone disease, it's a skeletal disease, as we see. This is a patient, Carol Orzel. This is the first adult I ever met with FOP. Carol was a wonderful individual. She came every year to speak to the medical students at the University of Pennsylvania and was very involved with FOP. And at a symposium 30 years ago, she saw the skeleton of Harry Eastlack. She wanted to see that. And we had the skeleton brought over to the symposium so FOP patients could see how this heterotopic bone forms. And she said to me, Dr. Kaplan, when my time comes, I'd like to be with Harry. I said, do you really want to be, do you really? And she said, yes, I do. She said, I don't have any family. My parents are deceased, and I have no brothers and sisters. And I would like to teach people in future generations about FOP. And her skeleton, this is her skeleton. She passed away several years ago. And according to her will and her wishes, her skeleton is at the Mutter Museum of the College of Physicians. It's in a closet with that skeleton of Harry, which she saw in life. She saw Harry. And we can see here the heterotopic bone. You can see that there's more bone formation in the dorsal regions. And if you walk behind the skeleton, you'd see dramatic sheets and plates and ribbons of heterotopic bone. And you can see the heterotopic bone more proximally than distally. You can see the heterotopic bone in the quadriceps muscles, and then posteriorly in the hamstrings, and that fuse the joint. So this is Carol's skeleton. And I have said that the key to the closet is the key to the kingdom. And this really says it all. So it is a true closet. The key to this closet is the true key to the kingdom. I would like to acknowledge the Nassau Professorship that sponsors the work that I do at the University of Pennsylvania, the Callie and Weldon Endowments. Their families, Ian Callie and Whitney Weldon, have FOP, and their family have generously given us endowments to pursue FOP research, and the International FOP Association, which has been a friend of ours from the very beginning. We kind of grew up together. There were 13 members in the IFOPA when we started. Now there are over 1,000. I've seen over 1,000 patients with FOP over the past 35 years. And so this is an ongoing saga. An ongoing adventure. And I haven't really described how we discovered the FOP gene. That I just described in the Grand Hamlin lecture I gave several years ago. But I wanted you to see today how the key to the closet is the key to the kingdom. The key to the closet is the key to the castle, looking inwards to these emergent properties, and it's the key to the kingdom of skeletal development as we look outwards to other diseases. Thank you very much for your attention.
Prof, thank you very much. And another perspective from the last time you presented in Shaykh Hamdan Award, and really kind of seeing what's applied for FOP can be used for good benefit for other people. Now, I have a couple of questions, but let me start with this. This regulation of PMB signaling. As you said, it's happening in embryo. What's your views that, because this is a rare disease, you usually don't detect enough. Do you think that we're losing some embryos given kind of this malformation in the skeleton? Is there a data on that?
Well, it's possible that even though FOP is an is an ultra rare disease that affects about between 1 in a million and 1 in 2 million individuals, so it's considered an ultra rare disease. The mutation that causes FOP is the same mutation in 97% of the individuals, in all of the classically affected individuals. And years ago when I spoke, a geneticist asked me a question. He says, why is this so common? I said, no, it's rare. He said, no, you don't understand the question. He said, FOP is rare, but this is one of the most common mutations in the human genome because all the mutations in FOP fall in the same place. So this is there's clearly some susceptibility there. I don't understand the what makes this region so susceptible, what makes this individual genes so susceptible. It's clear that the embryonic malformations don't cause lethality. Now, who knows? Maybe they do cause lethality of the embryos. We don't see in the animals any lethality in the embryo, in the embryos, male or female, or loss of litters, etc. In the animals that have, in the animals where we've in the mice where we made FOP. So I don't think it causes embryonic lethality, but it probably causes these malformations of the skeleton. And the children look normal at birth, except for this, as I said, this malformation in the great toe. I noticed many, many years ago when I was examining children that they had limited mobility of their neck, and they had limited chest expansion. Even before they formed heterotopic bone in these areas. And I said, why is this the case? And I looked at their radiographs, and I saw that they had intact joints, but a year later, those joints were gone. So, like I said, in embryogenesis, the increased BMP signaling pathway super inhibits the Wnt pathway, which is important for joint formation. As far as we can tell, there's no other embryonic pattern malformations with FOP. Now, it's very interesting. The FOP gene mutation is cannot be inherited in the mice. So all the mice that we made originally had to be made as chimeras, and they all have the malformations. The mice that we use in the experiments are all conditional mice. So we turn on the FOP gene late in embryogenesis, and this may be a problem due to the background of the mice, the genetic background of the mice. But it does give us, it doesn't seem to be a problem in humans, but it does give us a clue that increased BMP signaling pathway probably affects early embryogenesis, maybe not in humans, but maybe in mice, and maybe through different genetic backgrounds. And so it's possible that there is some embryonic lethality that we're not seeing.
Sure. That's great. Now, I had a question here from the audience saying, because there are spectrum of people from different providers, what kind of take-home message you want to give it to them to take on board for their practice?
Absolutely. The take-home message is that the study of rare diseases can often illuminate the study of common diseases. Many years ago, Dr. Roy Vagelos, who had been the chief executive officer of Merck Pharmaceutical Companies, he later became the chairman of the board of trustees of the University of Pennsylvania. And he came over to talk to me after he wanted to put me in touch with some other scientists, et cetera. It was a very heady experience for me as a young physician and scientist at the time. And he told me about the 5-alpha reductase abnormality, and he said, one of the major drugs that was developed for prostate hypertrophy was developed not from a rare disease, but from a disease that affected one individual. He said, he told me the story. He said, when I was at Merck, he said, we would have a journal club once a week. And he said, we went to the journal club and the light went off that this gentleman was infertile. This gentleman that was written in the case report was infertile because he didn't have a prostate. And 5-alpha reductase is important in the developmental biology of the prostate, for example. And scientists said, I wonder if we decreased 5-alpha reductase in people that had a prostate, that had prostate problems, and that led to the development of drugs for prostatic hypertrophy. So the message to the clinicians is not FOP. I'm using FOP as an example, as just an example. I think the real lesson is in the first part of the talk, that these rare diseases unlock common pathways, common conditions, and the reason is, as Porter and Fleshman said, nature is ultimately conservative. Nature doesn't make new genes, new cells, new pathways for common diseases. It's the rare diseases that show us the fidelity of these pathways. We wouldn't even know they existed. We wouldn't even know these phenotypes existed were it not for these rare diseases. So what I would say for the individual that's in private practice or in general practice, look to the rare diseases, not because they're rare, because they only affect a couple of people, and maybe you'll never see them, but they often give us the clue. They often lead us to understand the molecules, the genes, the pathways, the cells, the tissues that are involved in common diseases as well. And so that's really the lesson. The other lesson is these emergent properties. So because when you identify a gene, you're able to look inwards to that condition. You're able to use that gene. You know that is responsible for in some way responsible for the gene. Like in cystic fibrosis, for example, the chloride channel. So the chloride channel doesn't necessarily cause all the phenotypes or abnormalities of chloride channel in cystic fibrosis, but it allows you to investigate how that gene, the emergent properties of that gene in different cell types to understand how cystic fibrosis is caused, and that may lead you to understand various lung diseases, various pancreatic diseases, various other diseases as well. So these rare diseases, I think the lesson is that that the understanding of these rare diseases and the identification of the genes allows you to look inwards towards a disease, but also outwards to more common diseases. And that's really the lesson of rare diseases.
Crystal clear. Well, unfortunately, we have to end this because we have only 2 minutes on Zoom. Really, Prof, thank you very much. It was a pleasure to see you again, and education. And I leave the word for you to see you again in Dubai in person.
Dr. Al-Saleh, it is a pleasure. It is an absolute pleasure, and thank you so very much for that kind introduction, and really for inviting me back to Dubai. I know this is virtual. I had my wife and I had a fabulous time when we were there in 2018. It was a very warm welcome as I received today. So thank you so very much.
And I thank the audience for giving the time, making it a busy day, and to be here in the meeting, giving the importance of the talk and yourself. Thank you very much.